Okay, let's get started.
Hey folks, my name is Justin Angle and I'm an associate professor of marketing at the University of Montana College of Business. I also host a podcast called A New Angle. Welcome to tonight's installment of Alumni Virtual Engagement Series. This series is designed to keep you engaged and informed about the great things going on at the University of Montana, and it is brought to you by the Alumni Association. We are using a Zoom webinar system, so please use the Q&A feature on the bar at the bottom of your screen to submit a question at any time. The chat feature is turned off for this presentation. I will ask our presenter your questions after some brief opening remarks. Tonight, we are pleased to bring you my colleague, Dr. Jay Evans. Jay is a research professor in the Division of Biological Sciences and director at the Center for Translational Medicine at the University of Montana. Prior to joining the Division of Biological Sciences faculty, Dr. Evans worked in the biotechnology and pharmaceutical industry for 15 years at GlaxoSmithKline Vaccines, formerly Corixa Corporation, where he held various positions, including senior scientist, investigator, and project leader. While at GSK Vaccines, Dr. Evans worked on the discovery and development of novel vaccines, adjuvants, and immunotherapeutics. Dr. Evans completed his postdoctoral fellowship at the University of Texas Southwestern Medical Center and St. Jude's Children's Hospital, working in HIV immunology, stem cell transplantation, and lentiviral-based gene therapy systems. Jay, I hope I pronounced all these big words correctly. Please correct me if I have not. Dr. Evans holds a PhD in molecular and cellular biology from Oregon State University and a BS in biology from Pacific Lutheran University. Welcome, Dr. Evans.
Thank you. Thanks for the introduction. Sorry for some of the lengthy words in there. Lentiviral stuff and adjuvants are challenging, and I'll try to shed some light on what some of that means and what we're doing here at the University of Montana. You ready to get started through the presentation?
Let's do it. Share your screen.
All right. I will share my screen. All right, everybody can see that okay? Great, thank you. So thanks for the invitation to talk tonight about what we're doing at the University of Montana, and specifically what is the Center for Translational Medicine. We're gonna start off with just some general slides, outline about our group. You know, as you heard, we started from down in Hamilton, Montana at GSK and moved up to the University of Montana in 2016 to form this new center. We've got some exciting research going on in the new center, and I'll tell you a little bit about our coronavirus vaccine project. I'm only going to go through about 15 slides. I'm not going to get too detailed into the science of what we do to make sure there's lots of time for questions. So I know we've done a couple of these webinars, and there's usually a lot of questions about coronavirus and the vaccine and how long it's going to take and what we're doing to try to develop the vaccine. So again, we just heard the introduction. I'm going to talk a little bit about what is translational medicine and who is the University of Montana Vaccine Research and Development Team. And finally, I'll wrap up by telling you a little bit about our active vaccine research program on COVID. So what is the Center for Translational Medicine? This new center was founded in 2016 when our team moved up here from the University of Montana and recognized the need for a center that can help faculty and staff move new products and ideas from the lab to something that can help people clinically. And so the purpose of it really is to facilitate that collaborative interaction between researchers, not only at U of M, but across the Montana University system to translate those ideas, again, from the lab bench to the clinic. And the focus there is really to strengthen and expand the entrepreneurial programs available at U of M, with the goal of advancing technologies that have commercial potential. Right off the start of this, we had a whole bunch of faculty interested. Today, we have about 35 UM faculty that are part of the center with expertise everywhere from initial drug discovery and drug design through preclinical models of safety and efficacy and how do those drugs work, the typical types of things like toxicology and pharmacokinetics that are needed to get a drug to market, including expertise in GMP manufacturing of safe drug products. So really, we cover the space everywhere from discovery through IND. And we're also focused in intellectual property and licensing because if the university isn't patenting its inventions, it's hard to then develop those, form companies around them and commercialize those ideas. To put this in a little bit different graphical way is when you discover a new medicine or a new drug, it starts in basic research, oftentimes at a university like the University of Montana. It's a long process to get through the drug discovery process. And this question will come up is how can we develop a vaccine in 18 months or 2 years when the typical process takes 3 to 15 years to develop a new drug or a vaccine and costs over 800 million to 2.5 billion for every drug that's discovered. So where does CTM, the Center for Translational Medicine, fit into this? It's really in these early stages of basic research. When you have a new discovery or an invention and the university faculty member or staff wants to know how to get that into a company here in Montana, license it to a pharmaceutical partner, or move it towards testing in phase 1 human clinical trials. And so this is really our focus area, is to help move those inventions and discoveries at or other Montana University System campuses to that first stage of discovery where they get to be tested in people. So within the Center for Translational Medicine, there's a large group of us that are working on vaccine research and development. And so this started with 15 people, as mentioned earlier, that came up from GlaxoSmithKline down in Hamilton, Montana in January of 2016. Since then, we've grown to about 45 people on this one team working on vaccine discovery and development. We do everything from initial vaccine design through preclinical models of vaccine safety and efficacy, challenge models for the different pathogens that we might be looking at, candidate selection, process development, all the way up through the scale-up process development, manufacturing that takes to get something to phase 1. Some of the active programs we have in our vaccine discovery unit here at University of Montana. Of course, you'll hear a little bit about our SARS-CoV-2 project. We also have funding for influenza vaccine research, TB, Pseudomonas, E. coli, pertussis, which is whooping cough, Lyme disease, oncology vaccines, allergy therapeutic treatments, and an opioid vaccine program. So our vaccine program spans a lot of space. So it was pretty natural for us to step in and start working on the coronavirus vaccine pretty early after it was identified. One other important part about our research program is we have a lot of partners. And so this slide only shows our partners in the US. Of course, we probably should stop being so US centric because we have a lot of partners internationally as well. But as you can see, the University of Montana is connected with universities and companies that are working on vaccine research all over the country and really all over the world. Every one of our every one of our programs is partnered. One example of that is our coronavirus program. We are partnered with Boston Children's Hospital and Mount Sinai in New York to help evaluate our lead vaccine candidates that we're developing here at UVM. And one of our corporate partners locally is a company called Inimmune, which is a biotech company right here in Missoula. So whenever we talk about vaccines, we always have to do a little bit of history and what have vaccines achieved so far. Vaccines by far are probably the most successful medical invention of all time. Probably have saved more lives than anything else other than perhaps the clean water. For example, vaccines like the smallpox, polio, measles vaccines, the DTP vaccines, MMR vaccines, HIV vaccine. These have all been wildly successful, reducing the disease incidence by over 90% in most cases. And so, like I said, vaccines represent one of the safest and most cost-saving medical advances in healthcare history, probably second only to clean water. Each year, vaccines prevent 2 to 3 million deaths, and many children are also saved from disabilities. Just in 2016, this slide is a couple years old. It's estimated that 116 million children under the age of 1 received the DTP vaccine, for example, and that's how successful these vaccine programs have been. But despite all of these advances in immunization, about 1.5 million children still die every year from vaccine preventable diseases. So clearly we can do much better. The long list, anyone who has children knows there's a long list of vaccines you get in childhood. Some are available for travelers vaccines or special indications. But the success of these vaccines is hard to argue with. Things like measles and polio, which were once very common, now you very rarely hear about. With that said, this is what we have up against us right now. These are examples of emerging and re-emerging infectious diseases just in the last 10 years. It's kind of a scary slide, and you can see I actually added SARS-CoV-2 to this slide just today to exemplify this. So the question always comes out is MERS was out, we've had SARS before, why don't we have a vaccine? The question is, where do you put your money and effort and time in emerging diseases when there's so many things to work on? One thing that's for sure is this isn't the last pandemic or epidemic we're going to see. These come out on a regular occurrence, and it's anybody in the vaccine space. It's no surprise to any of us that we have a new viral pandemic that's affecting all of us today. I thought it might, if I was to guess, I would have guessed it would have been flu or Ebola, but it wasn't. It was a coronavirus. And so now the vaccine industry is really changing focus and changing gears and putting a lot of efforts on developing vaccines and treatments for this new SARS-CoV-2. And this is an example of that. I just grabbed this slide today. is part of the race to develop a vaccine against COVID-19, but we're definitely not alone. And this slide depicts that. There's over 500 compounds in development right now for possible treatments or vaccines for coronavirus. 135 different vaccines are under development, including the one at University of Montana. 160 some antivirals are being tested. Like 170 different kinds of compounds are being tested in clinical trials. So the effort that's going into trying to find a treatment, vaccine, or a cure for COVID-19 hasn't been seen before, at least not in my career. Yes.
Jay, can I interject with a question came in live about your previous slide asking what you mean by deliberately emerging diseases?
On this slide?
Yes.
What was the question again?
The question was, what do you mean by deliberately emerging diseases?
Deliberately, that's the anthrax one. Notice the only one on there is black is anthrax. That is a bioterrorism.
Okay.
Yep, that's what's meant by that. And so there are lots of things that are on the CDC list for possible bioterrorism agents, but that one is an emerging disease that was actually a bioterrorism threat from anthrax.
Super. Sorry to interrupt.
Sure, no, that's it. Sure, that's fine. And so this is a different way to look at this. The same is that the response to the COVID-19 is really unprecedented. The number of companies and academic labs and government agencies that are cooperating, the speed at which material transfer agreements and license deals are being signed to ensure the companies that are working on this have the resources and intellectual property and drugs that they need to advance them is, I wouldn't have guessed this could happen as quickly as it did. As you can see, in January, which was only 6 months ago, there was no research on coronavirus, nothing in the clinic, or very, very little in the clinic. And now you go out to May of 2020 and look at how rapidly antivirals and vaccines and other treatments have advanced. And you can see there where started its vaccine program on COVID-2. We were pretty early in the curve, and I think that's just a testament to the strength of the vaccine program we had at the time and being able to quickly change gears and the partnerships that we had as well. So what do we do at University of Montana when it comes to vaccines? Our vaccine platform is based on combining parts of the virus, what we call antigens, or the proteins from the virus, and immunostimulants that we've derived here at the University of Montana through our synthetic chemistry group. And those are called adjuvants. Those are things that we add to a vaccine to boost an improved immune response or a different type of immune response. Historically, vaccines have been inactivated viral particles or bacteria. They were very immunogenic. You can see by the blue bar there, they had really good immunogenicity, really good protection. Things like the whole cell pertussis vaccine falls into that category. From there, but these were also very inflammatory and very toxic, very reactogenic. They had a lot of bad side effects. So then the industry moved to subunit vaccines, toxoids, split virus vaccine like the split flu. A lot of the vaccines we have today are split or subunit type vaccines with still have decent immunogenicity, but have improved in their tolerability. In other words, they have less side effects. So the next generation of vaccines that are coming out now are based on purified antigens or pieces of viruses or bacteria that are completely devoid of all of the immunostimulatory features of that original virus or bacteria that also resulted in the high level of reactogenicity or toxicity. So what we're doing at the University of Montana is we take these purified antigens or proteins and we add adjuvants to them. These are synthetic compounds that we make here at the university that boost the immune response in very specific ways. And what I mean by very specific ways is we take something that represents the type of response your immune system might see if it might result in if you had a viral infection versus a bacterial infection. Those are very different types of immune responses. So we pick the right adjuvants or immunostimulants to match the type of immune response that we want to achieve. And so these combined adjuvants increase the immunogenicity of the vaccine, make it more effective without making it more reactogenic or causing some of the safety concerns that we had with the earlier non-replicating type of vaccine products. So how are we applying this to the SARS-CoV-2? This work was initiated here at University of Montana in February, like I said, in collaboration with our partners at Inimmune, Mount Sinai Medical Center in New York, as well as Boston Children's Hospital. Mount Sinai had previously worked on the SARS-CoV-1 vaccine and the animal models for that. So they were a natural partner. They have the biosafety level 3 labs with the animal models capable of doing that kind of challenge work that we can't do here at University of Montana. The group at Boston Children's Hospital, led by Dr. Ofer Levy, is specialty in vaccines and adjuvants that work at what we call the edges of life or the extremes of life, in the very young and in the very old. So they pattern these immunostimulants against peripheral blood cells, either from newborn or from the elderly, and find out which immunostimulants work best in those populations. To make sure that we develop a vaccine, we're developing a vaccine that can target the age range of population that we're trying to use the vaccine on. The vaccine adjuvants and delivery technologies we're using were discovered and developed right here at either University of Montana or Inimmune here in Missoula. So the lead vaccine candidates are currently being evaluated in animals at U of M. We've got at least 6 or 7 ongoing studies. Early results from those are very promising. We're able to dramatically improve the immune response against the SARS protein that we're using. The specific protein that we're using for a vaccine here is called the RBD domain of the spike protein, which is the protein, the part of the virus that interacts with the receptor on the surface of the cells that allows it to get into the cell. So if you can generate an antibody, a neutralizing antibody against that protein, you can prevent the viral infection. And a lot of other groups are taking the same approach. The unique part of our approach is the adjuvant and delivery systems that our group specializes in here at the university. The lead candidate vaccines coming out of this program will be moved over to Mount Sinai for testing in the viral animal challenge models. And we're actually at that stage right now. Our lead vaccine candidates are coming out. We just had discussions this week with our partner Florian Kramer at Mount Sinai about getting those vaccines over to him for testing in the challenge model. As soon as we demonstrate that these vaccines are effective in the challenge model, we'll then apply for funding to move those lead vaccines into human clinical trials. So as you can see, this is moving very rapidly. Not as fast as Moderna and some of the other companies that rapidly were able to move their vaccine candidate products into the clinic. It was partially based on the type of technology they were using. Either RNA or DNA-based vaccines are easier to manufacture and rapidly move into phase 1. Our technology is a little bit more traditional technology where we use pieces of the virus that have to be purified, combined with the right adjuvant, prove to be safety and the efficacy, and then we'll move those into phase 1. So my last slide, just talks about just our vaccine research program in general and kind of the future of vaccine research at U of M. I think the vaccine immunotherapy team here at U of M has a very bright future. Just since January 2016, we've brought over $45 million in new funding into these vaccine programs at U of M, including the COVID-19 vaccine program. Several of these technologies that we've developed over the last 2 to 3 years will be moving to phase 1 human clinical trials in the next 12 to 24 months, which is exciting for our group, also exciting for UM and the Missoula community. With that said, there's great opportunities for both undergraduates and graduate students participating in the research projects in our labs here at U of M. Like I said, we're about 45 people now. We expect we're probably going to need to hire 10 to 15 more people over the coming 12 months as we continue to expand. And so if people on the call have kids that are looking for a place to go to school, I know you're all UM alum, so you'll send them here anyway. But there's great opportunities for people interested in a career in science. And thanks. So I will take any questions, which I know there's a bunch. I know there were some that came in advance. I'm looking forward to. That's why we kind of didn't do a lot of science to make sure we could get everyone's questions and get them answered.
Super. Jay, we had a question come in during the session about antibodies. I'm going to read the question right out of the chat because I don't want to miss pieces of it. So the question is, some children have developed immune system overexcitability. Though testing negative for COVID-19, they demonstrate antibodies. For those who test positive for antibodies, are the antibodies specific to COVID-19, or might these antibodies be related to coronaviruses such as SARS and MERS and the 4 coronaviruses that cause colds, which infect people? If a child had experienced one of those 4 cold coronaviruses, would they be more likely to develop the immune system over excitability disease? When a vaccine is developed against COVID-19, how might the immune system over excitability disease be prevented?
That's a great question. I wish I had a clear, definite answer. Their publications are coming out. You can see the number of people working on COVID-2. These types of questions are key in the minds of researchers developing a vaccine. There's something known as ADE, or antibody dependent. Basically, it's antibody dependent enhancement of disease, where you can actually make the disease worse. It's been shown with dengue virus. It's actually been shown with SARS-CoV-1, which is why there's a concern with SARS-CoV-2. There is a chance that if you were exposed to a SARS-CoV-1 or a different coronavirus, then you have a suboptimal antibody response that's partially cross-reactive against SARS-CoV-2, you could have a worse disease symptom than someone who is naive to the virus. I haven't seen any publications where that's been shown for this SARS coronavirus 2, but it's key in the minds of everybody developing a vaccine, because you definitely don't want to develop a vaccine that is partially protective and could result in ADE. The animal models for ADE for SARS-CoV-2 are rapidly being advanced. The best model for that right now, I believe, is the rhesus macaque model, which nobody wants to have to test their vaccines in a primate model. It's expensive, and from an ethical standpoint, we'd rather stay out of primates in vaccine discovery and development. So hopefully either the murine or the hamster model will be able to develop a good model showing that antibody-dependent enhancement. In relation to the there's 4 regular circulating coronaviruses. There have been studies published now with both COVID-1 and COVID-2, looking at cross-reactivity of the antibodies across those different viruses. And COVID-2, some of the conserved domains, they did see some antibody cross-reactivity, but it was really, really low. So I expect, this is a hypothesis, that antibodies that exist against regular circulating coronaviruses probably will have little to no impact on immunity to COVID-2 or ADE, this enhanced response. But that's just a hypothesis on my part, since publications have not come out on that yet. That's a great question. It's something we think about a lot.
Super, thanks. So question from Bradley Marshall just came in, and this question that I think is on a lot of people's minds, you hear a lot about it in the press, is historically, how long has it taken to bring a vaccine to market in this similar category of disease?
Yeah, so you saw my earlier slide about translational medicine. I would say from the point where the vaccine is discovered, we have a new vaccine at U of M targeting a specific indication, that vaccine to commercialization is usually at least 10 years. If you were to rapidly advance the technology and have unlimited monies available to it, you might be able to reduce that down to 4 to 6 years. So if you had asked me this question in December, I would say a new vaccine, you better be 6 to 10 years. With that said, the coordination and cooperation that I've seen just in the last 6 months, and I've heard about with the FDA, EMEA, other regulatory agencies working with companies, BARDA, NIH, CDC, others working with companies, pharmaceutical companies, partnerships, universities to try to rapidly take down these roadblocks so we can get a vaccine to market quicker is pretty much unprecedented. The original estimation of, we think we can get a vaccine to market in 18 months. I certainly hope they can do it. It'd be the best interest of everybody if Moderna or one of the other companies can do it that quickly. I'm hesitant to think that they'll actually achieve that. I think 2 years is probably more reasonable, and that's 2 years from when they started, which was 6 months ago. So we might be getting in the window now where 18 months we might have a vaccine available, but that vaccine will likely be available for high-risk people for limited use because it won't have gone through the full barrage of phase 3 safety and efficacy studies as a normal vaccine would. So there might be a limited release, but anything would be better than what we have today.
Super. Somewhat related question that came in prior to us starting the live session here. It reads, news reports indicated that a vaccine had been developed or had been under development against the SARS-1 virus, but was halted for funding reasons. Though not identical, would any of the results from the SARS-1 virus be helpful in the development against the COVID-19 virus strains?
Yeah, absolutely. A perfect example of that is our partner at Mount Sinai worked on the SARS-CoV-1 animal models, challenge models there at Mount Sinai. So they already have and so now they're just adapting those animal models to use with SARS-CoV-2. And that's why as soon as we have a vaccine ready, they're able to test it in animal models. It was because of their previous work with SARS-CoV-1. And in the end, there were companies that had SARS-CoV-1 vaccines ready to go to phase 1, and then the funding dried up when the pandemic faded away. Of course, hindsight's 2020. Now we know we should have continued down that development path because it would have been possible to develop an FDA file around a pandemic viral vaccine for coronavirus, which would have really sped up the process of having one available now. Similar vaccines are available for pandemic flu through a couple of companies that could be stockpiled and are ready. And when a new flu virus comes out, they can then rapidly generate the antigen that's needed and move those into people much quicker. But again, you saw the list of different emerging and pandemic viruses and bacteria. Which ones do you target? Which ones do you fund? Because you can't do them all. But definitely the rapid progress that was made on this virus is in part due to research that's been happening with other types of coronaviruses or with SARS-CoV-1.
Sure. Jay, you've used the term challenge models a couple of times. It's a term we hear in the press as well. What is a challenge model?
So a challenge model is usually an animal challenge model. We like to use in vitro, what we call neutralization assays, and that is to see first in the lab in tissue culture, and what we call in vitro, if the antibody or type of response you're generating is able to block the virus from infecting cells. And that's a first step because the animal studies are usually the later step in the process. But ultimately, you want to know if a vaccine is safe and effective against preventing natural infection. So there's this SARS-CoV-2 virus, because of the protein on the surface of the cell called ACE2 that it uses to get in. For example, mice don't normally express ACE2. So mice aren't susceptible to coronavirus. Well, back when they were looking at CoV-1, they genetically engineered mice to express the human receptor. So now these mice became susceptible to coronavirus infection. Of course, right now, these mice are really hard to come by because everyone wants to do challenge studies with them. There's a Syrian hamster that's naturally susceptible to the coronavirus. And that's a model that's being used down at Rocky Mountain Lab in Hamilton, as well as several of our collaborators for testing vaccines and other treatments. And then ferret model has been used a little bit. They do get the infection, but they don't get sick. And then there's the rhesus macaque model. So those are the animal challenge models that are available for researchers that want to test a vaccine or a treatment.
Super. So you received a nice note from Jeff Stevens, who's a former visiting research science at Corixa. He said he did not have a question, but then he's chimed in with a question that I'll read next. COVID-19 infection appears to have unusual effects on the immune system, especially causing cytokine-like cascades in the lung, which causes many of the respiratory symptoms. As an immunologist, do you know of any information on the mechanism for that response?
Great question, Jeff, and hi. It's been a number of years. Hopefully you're doing well. So that's a great question. Most lung viral infections, when they have poor outcomes or morbidity associated with them, a lot of times it's not due to the virus, it's due to the associated immune system response and the inflammation in the lungs. Whether it's RSV or flu or coronavirus, lung inflammation is one of the big issues. Why that happens, why the immune system kind of goes over and above or gets out of control is something of constant research. There's a lot of people that are actually trying to develop anti-inflammatory medicines for the lungs or other parts of the body that will actually suppress the immune response to allow you to clear the viral infection, which would be counterintuitive to resolving an infection, but in later stages of infection might actually be the best thing. So we don't know the mechanism specifically, especially with SARS-CoV-2, but there's a lot of research into that. But it's a challenging model to try to replicate in the lab and to sort out exactly what's going on.
So maybe a quick question about your previous description of challenge trials. The question came in as you were talking about the animal trials about mice, and what is it about mice that they aren't naturally susceptible to this coronavirus?
They simply don't have the receptor ACE2 on the cell surface that the coronavirus binds to.
Okay. Let's pull the lens back a little bit. You've talked about the extent of your partnerships in the United States. Can you talk a little bit about how the world scientists are collaborating and sharing information on COVID-19? One would assume that there's some fast tracking of information. One would also assume that pharmaceutical companies want to corner the market first. Can you describe the environment as more collaborative or competitive? How's it feeling?
Yeah, no, I would say definitely there's more collaboration and cooperation that I've seen before in the vaccine space and even in the drug space. There's really an we had a coronavirus challenge that we just ran last week down at Rocky Mountain Labs in Hamilton. And the MTA process for us to be able to send them our drug candidate for them to test was a simple one-page document that we had done in a day, something that would normally have taken months of doing something like that with NIH. So the legal processes of how to share information have been streamlined, especially for coronavirus and rapid legal agreements. With that said, I mean, there's definitely a competitive nature to it, and biotech companies and pharma companies would love to be first to market because this would be a big vaccine product. But with that said, there will be significant government and social pricing pressures on anybody who comes up with a vaccine. So I don't think any of the, like I said, there's over 130, different vaccines in development. There might be a few, but I doubt any of them are doing it strictly from a profit standpoint. Because if they were, it probably wouldn't be the worth the investment given what the pricing pressures are going to be on this type of a vaccine when it comes out. So there is some competition. There always is in the space. But like I said, the level of coordination and cooperation between companies and government agencies is unprecedented. Nothing like I've ever seen before. And hopefully some of that sticks around long after the coronavirus is gone.
Super. Question about mutation. At this point, is the COVID-19 virus evolving or mutating quicker or slower than first predicted? What are the chances that this one evolves to a more virulent strain?
So that's a good question. So the mutation rate in coronaviruses, because they've been circulating for a long time now, is fairly well established and known. There's still some ongoing debate on it. One thing that's for certain, it does not mutate as fast as something like influenza. You see the seasonal influenza virus drift that happens, that one of the reasons you have to get a seasonal vaccine every year. It doesn't appear as though coronavirus is going to fall into that type of an RNA virus. It doesn't mutate that fast, which is great news for vaccine manufacturers and developers, and for anyone hoping we get a vaccine soon. There have been some mutations identified that appear to increase virulence, but it's more correlative. There was one that came out just a few months ago. In the US, a lot of the circulating strains have this specific mutation that wasn't found in the strains from China that appears to emerge when the virus spreads more rapidly or more widely in a population, but it's correlative. They haven't actually taken, as far as I know, they haven't taken that mutation and shown in tissue culture or animal models that it's actually more virulent. But it wouldn't surprise me. I believe they saw that with the MERS virus and the SARS-CoV-1, that as it moved through the population, it did gain some virulence as through mutations. Fortunately, this virus, like I said, doesn't have the high mutation rates of something like flu, which means a vaccine should be possible and on the horizon.
So follow-up question from Mark Pepler is there a chance SARS-CoV-2 could mutate and become resistant to the antibody your vaccine will raise?
That's a great question. If we were talking about a monoclonal antibody-based therapy, which they are doing, people are generating monoclonals that target the same spike protein for passive immunization treatments for people with SARS-CoV-2, then I would say yes. I mean, in a vaccine where we're targeting a whole protein with a lot of different epitopes, and more neutralizing epitopes in this virus are being identified all the time in publications, including more than just the spike protein. I would say with the mutation rate of this virus, the chance of it mutating away from the vaccine is much less likely than something like flu. It's always possible, but I think our opportunity for success here is greater.
It seems like another related question came in from Joanne as you were answering that. We know that influenza vaccines are based on the previous year. They have already identified at least 2 coronavirus strains. Will the vaccine only be able to target one strain and therefore become ineffective quickly?
That's a great question. There are 2 COVID-2 strains. They're calling them L and S strains that are circulating. And again, this is emerging data coming out every day. It's changing. The collaborators that I have that are working on this appear to think that a vaccine targeting one will likely be effective against both. But until they get efficacy data in human studies, no one will have 100% confidence in that.
Okay. Question that came in previously is, in developing vaccines for COVID-19, are researchers looking to minimize the chance of antibody-dependent enhancement? Mm-hmm. First, maybe define what that is.
Yep. So ADE, we talked about this just briefly earlier. Antibody-dependent enhancement is when you have a suboptimal antibody response against a virus that when you then get infected can actually enhance the disease. This has been seen in the case of dengue. There's 4 different subtypes of dengue. I'm not a dengue virus expert, so others on the call, maybe from RML, might know more about it than I do. But there's 4 different serotypes of dengue. If you only target one of those different serotypes, if you get infected with another serotype, your disease can actually be worse. You can actually, rather than neutralizing the virus, you can actually target the virus to infect other types of cells that it wouldn't normally infect, and cause disease enhancement. So this is a highly relevant and important question, because ADE has been demonstrated in animal models with COVID-1. I haven't seen a publication where it's been demonstrated yet with COVID-2, but everybody assumes that it would be the same case with COVID-2. So vaccine developers are critically focused on the safety and generating and advancing these animal models to the point where we can look for ADE, because we would much prefer to see in an animal model than in a human clinical trial. But I think with the slow mutation rate of COVID-2 and the fact that the neutralizing antibodies that have been generated to the vaccines to date appear to cross-react with the various circulating strains of virus, I think the chance of ADE is low, but it is a real risk that we need to take a careful look at.
Super. Let's switch gears a little bit. We have a question asking if there will be a second wave of the disease in the fall.
If I had the answer to that, I'd be on every talk show on on the TV. I assume there will. And the reason I say that is because coronaviruses, the other 4 coronaviruses have been circulating for a long time. There's coronaviruses that infect cats, there's coronaviruses that infect pigs. A lot of those other coronaviruses are seasonal in nature. Because of that, I assume that this one will be as well, and we will see it come back in the fall. Will be really hard to determine if that is coming back because of seasonal reasons or because of us opening back up and everybody going back to school. And of course, we won't have a definitive answer to that until several seasons go by. I'm assuming we'll have an uptick in the fall, though. That's complete hypothesis on my part, though.
Sure. So we got a couple of questions that have come in about a little bit of a hot button political topic, but Jay, I'd like to give you the opportunity to speak to it because you laid out in your slides the great number of public health benefits that vaccines have brought. We got some questions about the safety of vaccines, and you hear about the safety of vaccines, and it is an issue that's sort of become political. Can you speak to that a little bit?
I can. We talk about this a lot at work, especially since we're developing immunostimulants that boost the immune response to a vaccine. Anytime you're boosting an immune response, you have the chance of also boosting an unwanted immune response. So we talk about it a lot. So one thing I can say, I can't say all vaccine companies, every vaccine company that I've been a part of or interacted with have been focused on safety as their #1 priority. And it's such a high priority, it oftentimes gets in the way of vaccine development because they won't move something to clinical trials unless it has an absolutely squeaky clean safety profile in the animal studies. If there's even a hint of a safety issue, they don't move it forward, and really slows down vaccine research. So that I can say that. But one thing is for certain is that like any drug that's put out on the market, we have a very genetically diverse population. You can't predict how every single person, based on their historical vaccines that they've received, their infections that they've had, or their immune response. Some people have autoimmune diseases, hyperimmune diseases. You can't predict how every single person is going to respond to a vaccine. And like any drug, there are adverse events that happen when vaccines are given. And the goal is to minimize these to make the most safe and effective vaccine possible. But there's a lot of false information out there on the internet and webcasts about adverse events happening from vaccines, linking things like autism and other things to vaccination that's been proven over and over again to not be true. But vaccine safety is the paramount, and at University of Montana, one of the first things we do is look at safety in any of the new vaccines we're developing. And if they have safety signals that don't meet the profile we want, they'll never make it to phase 1.
Excellent. So you spent many years in the private sector before moving to the University of Montana. Can you speak a little bit about that experience and the differences between your life in the private sector versus at a public research university?
Yeah, no, definitely. And there's a lot of similarities, and there's also a lot of differences. My time at Corixa and GSK was great. I learned a lot about translational medicine and how to advance vaccines from the discovery stage to the clinic, which doesn't happen at universities very readily or very easily. But coming to a place like the University of Montana, there's been just an enormous amount of collaboration and doors that open because of how easy it is to collaborate and coordinate with people in a university setting. A perfect example of that is our coronavirus program. When NIH reached out to us and asked us to submit a supplement and work on a coronavirus vaccine project at the university, I made a couple of phone calls to Boston Children's, Mount Sinai collaborators we already had. We were able to get a project going in a matter of weeks and submit an application to NIH and get it funded really rapidly. That's harder to do in a big pharmaceutical company because of all the legal protections that have to be in place to protect their intellectual property and also prioritize the diseases and programs that they're spending money on. So the university environment has a lot of advantages, and it's really kind of a refreshing system for being able to collaborate and interact with people. The Center for Translational Medicine and our vaccine program at U of M here has expanded greatly because of those collaborations and how rapidly we can set those up. But if you want to develop a product to market or go through clinical trials, you really need to have a corporate partner or move that to a commercial space because someone has to fund the $800 million it's going to take to develop a vaccine through phase 3 clinical trials, and that's unlikely to come through the university system.
Super. Question sort of about how the virus handles different environmental changes just popped in. With many stating the virus can live on surfaces for short periods of time, etc. Can the virus live in extreme temperatures, both hot and cold, like higher temperatures in the Southern California desert in July, or colder temperatures like Montana in December? Any comments on how the virus behaves in different environments?
I really can't. There's probably literature out there that talks to that. I know the group down at Rocky Mountain Labs has done some environmental exposure, how long the virus lives on surfaces and things like that. They would be probably much better to fill the question than I do. I don't know. I wouldn't even venture to guess the heat, thermostability, or cold stability of what that virus might be. Other than I know we can freeze the virus because that's how we'll store it at ultra cold temperatures under specific conditions, though.
Okay, fair enough. So, question from Tammy about safety restrictions. And we talked a little bit about timeline before. We've talked about some of the political dimensions. Do you feel like safety restrictions might be relaxed as the virus, as there's incentives or pressure to develop the virus, the vaccine more quickly?
That's a great question, and that would be a better question for some of the, from the FDA or EMEA. What I do know from our previous interactions with the FDA is safety is number one on their mind. The last thing they want to do is put out a vaccine that's not safe, because that would set their agency and the vaccine industry back by many, many years. So I don't know for certain, but I'm assuming that the federal agencies are actually more focused on safety than they are on efficacy. Because if you put out a vaccine that's not safe, the pushback you will get from that and the negative press, not the negative press, the detriment that you could do would be huge. If you put out a vaccine that's not effective, at least you're not hurting people. Mm-hmm. So I'm assuming that if they do an early release of a vaccine like the Moderna vaccine, it will be after a phase 2 safety / small efficacy study, maybe even a phase 3 study. They may not know the full extent of how effective the vaccine could be in people that are very young or people that are very old, but they'll at least know that it's safe before they'll release it. At least that's my hope and my expectations based on previous interactions with those agencies.
Super. So you sort of answered the question about typical timelines for for vaccine development. Do you have any insights as to the timeline for development of other sorts of treatments?
Yes, oh, no, absolutely. The timeline for development of treatments, a lot depends on the disease you're trying to treat. I'll take one case. If you're trying to develop a treatment for a specific kind of cancer, you might have to look at 5-year survival to get enough efficacy data to even know if you can move it to phase 3. And then the phase 3 study, you might have another 5 or 7-year follow-up before licensure. So if you have timelines like that, they can extend the development pathway for by many, many years because of your efficacy readout. If you want to develop something for Alzheimer's or Parkinson's disease, sometimes the readouts on those studies, phase 2 or phase 3 efficacy, can be years. If you want to come up with a treatment to see if you can reduce the symptoms of the common cold, as you might imagine, you can get a lot done in a single cold or flu season, and you might be able to rapidly develop that kind of a treatment based on the course of the disease and how quickly you can get to the clinical outcomes you want to see. So
okay.
it could be anywhere. You know, the one drug might take 15 years because of the type of disease or indication you're going after. Another one might be 5 years from phase 1 through phase 3.
So outside of development of a vaccine or treatment, do you have any insights as to the supply chain for vaccine production and the current state of affairs in that space?
So that's another great question. So each vaccine has its own supply strain constraints and issues based on the nature of that vaccine. I believe that in the case of the one vaccine that went to the clinic first, which is being developed by a company called Moderna, it's an RNA-based vaccine packaged in a lipid. It's a novel platform that they and a couple other companies had developed a number of years ago. In that case, no one has manufactured an RNA-based vaccine on the kind of scale that we're talking about, which is, I believe, a large part why BARDA, I believe it was BARDA, invested like $480 million in Moderna. It wasn't just for their rapid advancement of their product through phase 1, phase 2, phase 3 clinical trials. It was so they could start scaling up their manufacturing capacity now. Because if their vaccine is successful, no one's ever manufactured that type of vaccine on that scale. I have no knowledge of whether they're going to be successful or not. I certainly would hope so.
Super. Okay. Let's see. So, question about other vaccines. So I understand there is some correlation data coming out of Hong Kong about the MMR booster shot and recovery from COVID-19. Is there anything promising there?
So that's an interesting story. So there have been just recently, the last couple of months, a couple of publications that have come out. The first one came out linking MMR vaccination and lower mortality and incidence rates for COVID-2. Which is interesting. They targeted some populations, a couple of places where the more recent MMR vaccination campaigns were done, and they had really low mortality rates. Second to that, about 2 weeks later, another publication came out showing that the rubella vaccine, which is part of the MMR, there's a protein in there that has about 29% homology to the same protein in the COVID virus. So it's correlative, not necessarily confirmed that that's the case, but there is some promise there. The other thing to note, though, is that there have been reports of, for example, the BCG vaccine, which is a vaccine for TB, might also have positive outcome effects on several viral infections, including COVID. So could these patients have gotten the BCG vaccine and the MMR vaccine? And that's why, potentially this, what we call, it's hard to explain, but it's called trained immunity or trained innate immunity. It's a different type of immune response that allows your your body to respond to a bunch of different types of pathogens. Is if that's what's going on there, or there's true cross protection between MMR and COVID is yet to be seen. There's clearly more research is done on this. It would be a pretty exciting development. And the other thought is the MMR vaccine came out in the early 1970s. If this ends up being true, it could go a long ways towards explaining why the elderly are the most susceptible. Of course, that's yet to be, that's pure hypothesis on my part, but there are a couple of reports suggesting there might be a link there.
So we got a couple questions, Jay, about testing. There's been much discussion regarding COVID testing, including antigen testing and antibody testing. Could you speak about the differences between these two types of testing, and when each one is used, and how testing can translate into public health policy and initiatives?
No, I can, obviously the testing is not what we work on at the University of Montana, our group, but we're very familiar with it. So the two main types of tests that are available, one is you hear about called the PCR test or the viral test, and that's where they're actually looking for virus, whether it's in saliva or in sputum or in your blood or wherever they're going to look for it on a surface, on a contact surface, looking for survival of the virus in the air. And that's a test called PCR. It's a polymerase chain reaction where they actually amplify part of the viral RNA and see if they can detect it. If it can't be detected, then it's negative. If they come up positive, they confirm the result, and they can say you have a certain number of viral copies in that saliva sample or sputum sample. And that's only going to work when you're actively infected with the virus. That 5 days or 7 days or 10 days when you have an active infection going on, you could potentially expose other people to the virus. That's when that test works. It can be a fairly rapid diagnostic test. The other one is a serology test or an antibody test, and that looks for antibody in your bloodstream against the virus. That test also can have some false positive because, like we talked about earlier, some of the other coronaviruses do have some cross reaction with the SARS-CoV-2 as far as antibody. So they're working out some of the false positives that they had seen in that test. I think there's some tests out now that are giving very good results. So that tells you if you've been exposed in the past to the virus, not whether you actively have the infection. And so both of those have different purposes when it comes to public policy. The first, the PCR test, is probably the most useful because it tells you who in the community actively has circulating virus. But just because you tested negative this week doesn't mean you can't test positive the following week. But it can help rapidly identify an outbreak and do that contact tracing just to figure out who the infected patient might have exposed. The serology test is more of a system-wide test. How many people in a community possibly have been exposed to people who were non-symptomatic, and then might subsequently be immune to follow-up exposure to the virus. I have to mention, there is a company right here in Missoula called Fire Diagnostics, which is actively working on development of a PCR test. And they've had a lot of success, and they have a specific testing method that hopefully will get through some of the bottlenecks in the current PCR and base method and some of the equipment that's required, and they've had a lot of success. That was a company that also came out of the University of Montana.
Jay, can you speak to some of the efficacy or lack thereof of existing medications, and if they've been tested on COVID-19?
I would need some more clarity on what you mean by existing medications.
Yeah, so the question tests me because it's got a bunch of words I don't know how to pronounce. So what are some of the existing medications already used for cytokine cascading ailments? And have these been tested against COVID-19?
Oh, okay. That's a great question. I mentioned earlier that in some cases what you're trying to treat is symptoms, not the virus itself. In the case where you have an overactive and inflammation response, whether you use steroids or things like that are commonly used to suppress the immune system, whether those would be effective at reducing the cytokine storm and preventing some of the morbidities or mortalities associated with COVID-2. I'm assuming some of the 500 drugs in development are in that class. I have not heard of any success in that case. Most people are trying to identify antivirals or vaccines to prevent the virus itself rather than treating the symptoms.
And one last question. How do you volunteer to be a part of a trial?
That's a great question. It's a very complex system. Usually phase 1 safety studies are single site studies, and if you're not at the medical center or clinical site that's running that phase 1 study, it'll be hard to even find it. They'll advertise locally in that market for it. Once they get to phase 2 studies, usually they're multi-site studies. Sometimes 2, 3, 4 sites will be recruiting patients, and you would have to be close to one of those sites and hear about it and be recruited into that cohort for that phase 2 study. And then phase 3 studies have thousands of patients in multiple sites. And whether there would be a testing site in Missoula for a phase 3 clinical study would just depend on whether community or St. Pat's was involved in that clinical study.
Super. Well, that is all the time we have. Dr. Jay Evans, thank you for joining us and sharing your wisdom and experience and helping me with the bigger words. Please join us next Wednesday when our colleague, Dr. Michael Cassens, who is the Director of Esports at the University of Montana, will join us. He'll be talking about our new esports team using gaming practices and theories in teaching, and he will also have 2 members of the inaugural esports team along to join him. You can also check out my latest podcast episodes at anewanglepodcast.com. On behalf of the University of Montana Alumni Association, thanks for attending. Thanks for your questions. Good night. Take care and go Grizz.